I want to thank the Nobel Committee on Chemistry for recognizing ion channels in 2003. My contributions to the field of ion channels has relied very heavily on work of other scientists who came before me. Since the days of Alan Hodgkin and Andrew Huxley, who first showed that the nerve impulse was propagated based on ion conductances across cell membranes, electrophysiologists have used techniques to establish the existence of ion channels and has given us a rich description of their function. Molecular biologists have identified the ion channel genes, and structural biologists have developed and advanced the techniques for protein structure determination. Without all of this background, I wouldn't be here today to talk about ion channels. I'm very fortunate to have come into the field of ion channels when I did. The subject of my talk today is here, the atomic basis of selective ion conduction in potassium channels, a subject that's very dear to my heart. And I've prepared this lecture for the most important people in the audience, and that is the students of science, and I hope that I've done a good job. The fundamental issue is this. All cells are surrounded by a thin, approximately 40 angstrom membrane called the cell membrane. And the cell membrane is essential for holding the chemical components of life in one place so the chemistry of life can happen. But the membrane is a barrier to substances that have to cross from inside the cell to out. In particular, it's a barrier to charged atoms, that is ions, like potassium, sodium, calcium, and chloride. And the reason that the membrane is a barrier is because the inside of it is an oily-like substance. And we know that ions prefer to be in water than in oil. And I demonstrate this here in a simple way by taking the salt cobalt chloride and shaking it in a mixture of oil and water, and then letting the phases separate, and you see that the red cobalt and chloride stays exclusively in the water phase on the bottom. And the reason that that happens is because polar water molecules interact favorably with an ion, as depicted in the cartoon here, with the cation in the middle and the negative ends of the dipolar water against the ion. The cell membrane is the same way. With its oily interior, an ion will tend to stay on the water on either side of the membrane rather than go through the membrane. And so cells needed special systems for letting ions cross the membrane. And in nature, we discover two general kinds of systems. It's been known for a long time, the pumps and the channels. Now, the pumps are proteins that concentrate ions across the membrane. They pump them against the gradient, in fact, concentrate them, and they use energy from another source, such as ATP hydrolysis, to do this. So the pumps concentrate ions. The channels, on the other hand, are passive devices, and that means when they open, the ions run downhill. They go from a high concentration to a low concentration. Now, one way you could describe this combined system of pumps and channels is to say that the pumps store energy across the cell membrane in the form of ion gradients, and the ion channels use that energy for useful purposes for the cell. For example, here, a picture of a cartoon of a potassium channel letting green potassium ions through it. When it's open, allows the potassium ion to run from inside the cell, where it's at a high concentration, to outside. But when the potassium ion crosses the membrane, because it has a +1 charge, it moves a charge across the membrane. And this leaves the membrane negative on the inside and positive on the outside. It electrically polarizes the membrane. And this electrical polarization of the membrane is at the very heart of electrical signaling in biology. Now, notice one thing, that if this potassium channel allowed the red sodium ion to go through, it would run down its concentration gradient in the opposite direction, and it would cancel the effect of potassium moving out, and it would cancel the charging effect, and the system would fail. So what that means is an ion channel not only has to conduct ions, but it has to do so selectively. Now, potassium channels are exquisitely, they're remarkable molecular devices in that they conduct potassium ions at an extraordinarily high rate. At a rate of, and under electrochemical driving forces typically found in cells, potassium conducts at a rate of 10 million to 100 million ions per second. How fast is that? That's about as fast as potassium ions can diffuse from solution up to the pore. So they go through with a very low resistance. What's more remarkable is that the potassium channel selects for potassium over the smaller sodium ion with a fidelity of about 10 to the 3 to 10 to the 4th, 1,000 to 10,000. So it essentially lets potassium ions through and does not let sodium ions through. Now, this seems to raise a paradox, because you could say, how do I get a very selective potassium channel? And you could say, well, to get a good potassium channel, I need to make a pore with binding sites that are just right for binding potassium. But then if you think about it, if those sites are just right for binding potassium, the potassium should be very happily bound there. And then you ask, how does the ion go through so fast? How do you get such high rates? So high selectivity and high conductive rates seem to be counter to each other. And how did nature solve this problem? I got interested in this question when I first went to work at Brandeis University in my postdoctoral work with Christopher Miller. And there, I began to work with, of all things, scorpion toxins, small proteins from the venom of scorpions. By making electrical measurements, Chris Miller and I found that these toxins inhibit potassium channels by plugging the pore, as shown here in a picture from a paper at that time, the toxin acting as a plug. Now, this was a very humble conclusion, actually of interest only to a few biophysicists at the time, but the timing was right, because shortly after this study, the first potassium channel genes were cloned. This was the shaker potassium channel gene. It was cloned in the laboratories of Lily and Yunung Jan, Olaf Pongs, and Mark Tanaway. This was a very important advance because it gave us the first amino acid sequence for a potassium channel. But it didn't tell us which of those amino acids, for example, made the pore or any other part of the potassium channel. But we knew that these pore-blocking toxins would allow us to home in on the pore and find out which amino acids made the pore and also learn other things about the channel. Now, work for several years with these toxins taught us many things about potassium channels. I'll just briefly tell you two. One was, it told us that potassium channels had to be tetramers. And this experiment came from actually mixing wild-type potassium channel subunits with mutationally altered subunits, altered so that they wouldn't bind toxin, and then studying the sensitivity of the resulting population. And by making the right measurement and having a knowledge of the binomial distribution, you could determine the subunit number. For example, on this graph, this quantity plotted here should go to the subunit number as you go to high toxin concentration. And what you can see is the data points are approaching the number 4. So that told us there must be 4 subunits in a potassium channel. Now, this wasn't a very surprising conclusion because sodium and calcium channels that had been cloned in the laboratory of Shosaku Numa had 4 repeats, and each repeat looked very much like the subunit of a potassium channel. But it did give us this picture of 4 subunits surrounding a central ion conduction pore. The next early result I want to tell you about is that the selectivity in a potassium channel is conferred by a segment of amino acids that we called the P region. This is a linear representation of a shaker potassium channel subunit. The dark segments correspond to membrane crossing regions. And in this region here between the S5 and S6, I show in single letter code an amino acid sequence. And what we found through mutations is that two clusters of amino acids marked with asterisks, here and here, interacted with the scorpion toxins. So we knew that these had to be near the external entryway to the pore. And then, working with Gary Yellen, using an inhibitor called tetraethylammonium ion that blocks potassium channels from the inside, we found that amino acids in the middle, one marked here by an arrow, actually is on the inside. Tetraethylammonium ion, or TEA, was a well-known blocker of potassium channels. Clay Armstrong and Bertil Hille had used it decades earlier to actually demonstrate the existence of potassium channels in cell membranes. And Clay Armstrong showed that this inhibitor acts by entering into the pore and plugging it from the inside. So with these results, we knew that this segment had to be outside, and then this part inside, and then this part outside. And working in this region, my first graduate student, Lise Hegenbotham, and postdoctoral scientist Jie Lu made mutations and found that amino acids marked with a plus sign affected the ability of a potassium channel to discriminate between potassium and sodium. So we knew this was a very important region. And so around this time, we had a rough picture of a potassium channel like this, as a tetramer, here 3 subunits shown, with P regions reaching into the center to make a selectivity filter. And these amino acids that were responsible for selectivity turned out to be very interesting because laboratories throughout the world began to clone different potassium channels. Many of these were very different from the Shaker potassium channel, except in one respect. They all contained the P region amino acids, especially the ones important for potassium selectivity. And in fact, this sequence became the hallmark for identifying potassium channels out of gene sequences. And here I show you this sequence that we called the potassium channel signature sequence from organisms throughout the tree of life, from bacteria to humans. And you can see that this is highly conserved, the single letter code, small variations on a single theme. And this high degree of conservation told us one thing. It really meant that, early on in the evolution of life, a solution for selectivity for potassium ions in a channel must have evolved, and then nature stuck with that solution to have it be so invariant across the tree of life. Now, at that time, I thought it's one thing to know where the sequence is and know what the amino acids are that are doing the job, but it's a very different thing to know how they do what they're doing. And I knew that we would never know what they're doing unless we could see the structure that these made. And in fact, at that point, I decided we had to become crystallographers. And in fact, just as a message to young scientists, students in the audience, that if you reach a point where you really want to know something, and it might involve doing something that you know you have no business doing because you don't know anything about it, you should do it anyways. Because I think it's better to try and fail than never try at all. Because that first path of trying is the only way you'll ever really do it. So, trying to figure out the atomic structure of a potassium channel and how these amino acids do what they do really has consumed my time for the past 8 years and the time of my student and postdoctoral colleagues who have come to work with me. Potassium channels, we now know, are divided into 3 general classes. Here I show a single subunit representation of the different classes. And remember, 4 of these make the potassium channel. The simplest ones consist only of a pore, and that has a membrane-crossing helix, a pore region, and an inner helix. And the more complicated ones have the same thing, the same pore, but they have additions that have to do with the channel's ability to gate open in response to a specific stimulus. I'll come back to these more complicated channels in a little while. The simplest kind, that consist only of the pore, seem to be the right kind to try to see how the potassium conduction process works. And one from a bacterium, Streptomyces lividans, its signature sequence shown here in the top, called KCSA and identified in Hilgun Schrempf's lab, seemed to be the best candidate to pursue to determine the structure. And after a focused effort, my laboratory determined the structure of the Shaker potassium channel after finding crystals that would diffract X-rays well enough. And there were crystals of the type shown here in this picture taken at a synchrotron. Now, this first structure was to 3.2 angstroms resolution, and it took 4 additional years to identify crystals that would diffract X-rays to better than 2 angstroms resolution. And here's a diffraction pattern from those crystals. And this defined the chemistry of the potassium channel at the level we need. The blue mesh is electron density, and you can see in here potassium ions held by the protein. And this is what we were after, this kind of understanding. Now, this effort has been a long effort, and it's been a team effort in my lab. But I want to mention a few key postdoctoral scientists who have worked on this. One is Declan Doyle, who worked on the initial structure from the beginning until the end of it. Another is João Marai Cabral, who worked on the initial structure and then worked on deducing the mechanism of the ion throughput that I will tell you about. And another is Yufeng Zhou, who finally determined the high-resolution structure, determined the absolute occupancy of ions in the selectivity filter, and figured out a new principle for high-throughput rates that I will tell you about. This is the KCSA potassium channel with its 4 subunits colored in different colors. The top is on the extracellular part is on the top, the intracellular on the bottom, and the ion conduction pore runs down the middle between the subunits. This you can see better in a simpler representation of the channel, with 2 subunits removed, and we can see just 2 subunits with the pore down the middle. The blue mesh shows ions and water lined up along the ion conduction pore. And when we look at this, you can see that the ion conduction pore is very wide in the middle of the membrane. It's wide enough so that an ion there is surrounded by water. It remains fully hydrated. And this structure has interesting helices that I've painted in red here, called pore helices. They're pointing the C-terminal end of these alpha helices towards the center of the channel. And each subunit points one there. You can see 2 here. The C-terminal end of a alpha helix is associated with a partial negative charge. And remember, potassium has a positive charge, and you have the negative end of an alpha helix pointed at it, and that makes sense to help stabilize it. Now, remember in the beginning I told you the fundamental issue of the cell membrane is an ion doesn't want to enter the oily substance in the middle. But what we have here, you see in the structure of the channel, is a pore that allows an ion to remain hydrated at the center, deep within the membrane, and it points partial negative charges at that ion to stabilize it. And so this makes sense for allowing an ion to cross the membrane. Now, what makes this a potassium channel is the selectivity filter, shown here in yellow. Here, potassium ions are held in a dehydrated state. This structure, the selectivity filter, is made by those signature sequence amino acids, and we'll look at a closer view of it here. What you see here are carbonyl oxygen atoms, shown as red sticks, all pointed in one direction from each subunit at the center of the pore, and they interact with potassium ions that bind at 4 positions, labeled 1 through 4, from the outside. These carbonyl oxygens act as surrogate water molecules to hold the potassium ions. Looking at this structure, we can tell why nature has conserved this sequence. The glycine amino acids are special because they easily sit in the left-handed helical region of the Ramachandran plot with their dihedral angles. And so we effectively have LD-LD amino acids here. a sequence of alternating L and D amino acids, and that's what allows the carbonyl oxygen atoms to point to the center to interact with the potassium ions. The hydrophobic valine and tyrosine side chains point directly away from the center, and they're packed in a hydrophobic core to give geometric constraint to this structure. Here we'll look at another representation of this. This is the selectivity filter with the 4 ion positions. If we look at a potassium ion in any one of these positions, a potassium ion sits in a cage surrounded by 8 oxygens, 4 on the top, shown as little red spheres, and 4 on the bottom of this potassium ion. And so it sits in the center of a cube of oxygens from the protein, or a twisted cube called a square antiprism. Now, a hydrated potassium ion that sits in the wider part of the ion conduction pore below the selectivity filter has water molecules around it, and it shows us something very beautiful. The potassium ion with water around it has 8 and has 4 on the top and 4 on the bottom. And if we now compare a potassium ion in the selectivity filter with a potassium ion in water, we see why this is a potassium channel. You see that the protein here has made sites that mimic the hydration shell around a potassium ion. That's what nature has done with this structure. And this selectivity filter allows ions to move from one site to the next in an environment that is like water, but it is the protein. These sites are the right size for potassium, and you might ask, what happens if I try to take the potassium out of this structure and replace it with the sodium? If you do that, what you discover is the structure doesn't maintain itself. Here is the structure we're looking at in the presence of high concentrations of potassium, with the ions in 4 positions. Here is the structure in 3 millimolar potassium and about 150 millimolar sodium, replacing the potassium. It looks different. You don't see 4 positions. I'll come back to this structure. The point for now is it's different. We call this a collapse structure. And what this means is, in fact, the selectivity filter is truly tuned for potassium such that its structure requires potassium to be maintained. Okay? Now, when we look at this, we see 4 positions. But actually, if we ask, what is the occupancy of each of those positions, you find that the occupancy is approximately 1/2 at each. That means half the time a potassium ion sits there. The reason you see 4 peaks is because in a crystallographic experiment, this is like a superposition of photographs. So you see an average position of where the ions are. On average, there are 2 potassium ions in this selectivity filter, distributed over 4 sites. And in fact, from experiments that I don't have time to tell you about, we know that those 2 potassium ions tend to sit in 2 specific configurations that I show you here. Here is one picture, potassium as green and water as red. So the configurations 1, 3 are potassium, water, or water, potassium. And what we're looking at here is kind of a combination of these 2 configurations. In fact, one hint for that I said is that if you look at the electron density just above the selectivity filter, you see that there are extra potassium ions out there. And in fact, a cation is drawn to this region of the protein in the crystal because it's very electronegative. There's a lot of negative groups up there. But it doesn't sit in one energy minimum place. It's actually distributed over 2 positions that are spaced equal to the spacing in the selectivity filter. And an explanation for that is that the potassium ion out here actually feels the distribution of ions in the filter. And in the 1, 3, it's pushed further away. And in the 2, 4, it's drawn further in. Okay? But these 2 configurations, 1, 3, and 2, 4, it's interesting because they sit at the ends of a very simple throughput cycle that I show here in a simpler representation of the filter, where the potassium ions are green, 1, 3, with water in between red. 1, 3 configuration can just go over to the 2, 4 by the queue of potassium and water just moving, shifting in this direction. So they can hop here and hop back through thermal agitation. They would imagine they would move back and forth. But imagine when you're in the 1,3 position, a potassium ion can enter from underneath and move the Q as an ion exits from the other side. And so a complete turn around this cycle, one turn corresponds to one ion going through. And in fact, this is just a little cartoon to show you what I mean. I don't show the waters in between the potassium, they would be implicit here, but you'd have thermal vibration of the ion water Q in the selectivity filter, and then you'd have movement of an ion, and of course this is a completely passive device, so the direction of the cycling is dictated completely by the electrochemical driving force of the ions. It can go in either direction. But the point is, you always have two potassium ions in that filter, and a third one comes through, and one pops out the other end. Now, remember in the early on, I raised a paradox, and that is, how did nature come up with a device that has high selectivity and high throughput at the same time? And I'd say one of the explanations for this is that if you look at the filter, it has ions in it that are very close together. It has multiple sites and ions that are closely spaced. The mean center-to-center distance between potassium ions in the selectivity filter is just a little over 6 angstroms, 6 to 7 angstroms. They're quite close. They both carry a positive charge, so they'll tend to repel each other. So I would say that an individual site would have a high affinity for potassium, but because you have ions close to each other, the ions repel each other, which will lower the affinity. So that's one of the explanations for why you could have high throughput, why the ions won't stick too tightly, and you'll have high throughput in the setting of high selectivity. But there's another explanation, and that has to do with this structure of the selectivity filter that I showed you in the presence of low potassium concentrations. This is looking again at this collapsed selectivity filter in potassium concentrations less than 5 millimolar. What you actually have here is, on average, 1 potassium ion distributed over 2 sites, and the filter is a little bit hourglass-shaped. In fact, it's pinched kind of shut here in this collapsed configuration. And this is the filter we've been looking at, the conductive form of the filter, in the presence of high concentrations of potassium. So the filter in low potassium concentrations is here, and in high concentrations, it's here. Now, certainly, when ions are conducting rapidly through this device, it's not undergoing this conformational change. That's because, one reason is because when a potassium channel is opened and conducting, ions are going through at a very high rate, and the potassium concentration inside the cell is very high. So you're in this regime, or this condition of conduction. So in a sense, you could say this collapsed state of the filter is a laboratory phenomenon. You know, it's something we see in the laboratory when we lower the potassium way down. But in fact, this laboratory phenomenon has meaning. It's telling us something about the selectivity filter, that it's doing a trick, actually. It's made to do a trick to ensure that ions don't stick too tightly. And let me tell you why I say this in a little thought experiment. Imagine if the selectivity filter maintained its structure, even when we lowered the potassium concentration, so that it maintained its conformation for conduction, even when there's only 1 potassium ion in it. Then, when going from the 1-ion state to the 2-ion state, you could say that the free energy from left to right here would just be given by the binding energy of this second potassium ion. Simple statement. This would be a hypothetical case. It's not what we see. What we see in cartoon form is this. We see that the 1-ion state has an ion either here or here, 1 ion, and it has a different conformation, and when a second ion binds, it goes to here, to have 2 ions in it, and it achieves its conductive state through a conformational change. Now, in this case, the free energy in going from the left to the right would be the free energy of the ion binding, just like up here, minus the work it took to do the conformational change to make the selectivity filter like that. And the energy for that work came from somewhere. It would have to come from the binding energy of the ion, which means the ion does not bind as tightly. And so this is a way to ensure that the ions wouldn't bind too tightly. This explanation matches exactly an explanation that enzymologists came up with for enzymes some decades ago in trying to explain how you could have an enzyme that is so specific for a certain substrate and yet have a Km that is something like the affinity that's very high and appropriate for the cell. And they called it induced fit. And in this case, I would say something very similar to what happens in enzymes is happening in the selectivity filter of the potassium channel. So I want to summarize the principles of high selectivity and high conduction rate by saying that there are 8 oxygen atoms surrounding potassium ions that form a selective coordination shell at each site. That sequential sites bind 2 potassium ions in either 1,3 or 2,4 configurations, and 2 ions in close proximity tend to repel each other and favor rapid conduction. And to enter the 2-ion conducting states, a conformational change occurs. This change will cause potassium to bind with lower affinity and conduct more rapidly. Now, the main focus of my talk is on the selective conduction of potassium, but I want to say something about channel gating. That is, the process by which channels open and close. Here on the bottom, I show a channel record, an electrical recording of a channel undergoing doing what it does. And what is graphed here on the y-axis is current, and on the x-axis is time. And what you see in a record like this is the current will remain sometimes at a 0 level, and then it will jump up to a non-zero level, and then you'll see it go back down, and it will come up and down. What you see is the current is turning on and off. And what we say is the channel is opening and closing. And we have in mind a picture like I've shown here. A closed potassium channel with a gate or a door covering its pore. And then an open potassium channel that is conducting ions by a mechanism that I've been talking about. And the switching between on and off would correspond to the gate opening and closing. Now, what's really happening here? How does a potassium channel open and close? This is back to the classification of potassium channels, and I told you that more complicated potassium channels, they all have a pore, like the one I've been talking about, but they have additional protein components added to them. Ligand-dependent channels usually have protein added to the C-terminal side of the pore, and voltage-dependent channels have extra protein added to the N-terminal side of the pore. And this extra protein allows the channel to respond to a specific stimulus so that it opens under the right condition, in a sense so that it opens in the cell when it's called upon. Now, quite a few years ago, we decided we want to see what these more complicated channels look like, to try to understand gating, the structural basis of gating. And again, this has been a team effort, but there's one postdoctoral scientist that I want to mention, Yuxing Zhan, because he's actually determined the structure of both a ligand-dependent ion channel, this kind, and a voltage-dependent ion channel, this kind. These two channels are called KVAP, the voltage-dependent channel, and MTHK, a calcium-dependent channel. The ligand is calcium. Now, the KVAP is from a thermophilic archaea. MTHK is from a thermophilic bacterium. Despite the high temperature prokaryotic origins of these channels, they have counterparts or channels that are very similar in eukaryotic cells. The gating response to the stimulus is shown in channel records here. For KVAP, you can see the voltage dependence of gating if you have the channel in the membrane and you hold the membrane at a negative voltage inside with an external amplifier and then depolarize the membrane, what you see is the channel is closed at negative and that it opens for a while. It tends to open when you depolarize the membrane or make it positive on the inside. And these are repeated traces. And you can see that it's the stepping of the voltage that makes this channel open. And this process is related to this called a voltage sensor, attached beside the pore. The voltage sensor is a helical structure. It has positive charged arginines on it. And we have a long way to learn about the details for how this is working. But what we know is that the component with the positive charges move a long way through the membrane in association with membrane depolarization to open the pore. This gating stimulus for the calcium-dependent channel is shown here. In the absence of calcium, the channel is open very infrequently, but then if calcium is raised inside the cell, the channel opens more. And in fact, at high concentrations, you could see that there were 2 channels in this membrane. So in this case, it's calcium that's causing the channel to open. And what causes that to happen is this large component, this calcium sensor, attached not in the membrane, but on the cytoplasmic or intracellular side of the channel. And let's now take a closer look at that in this picture. Here is the pore of the potassium channel. So this would be in the membrane. And here is the calcium sensor. We call this big thing a gating ring. And for a technical reason, in the structure, the electron density connecting the pore to the gating ring is not well defined, so I show it as dotted lines here. And we, again, like in the voltage-dependent channel, there's still much to be learned about how this works. But the general idea is this, that the gating ring changes its shape when calcium binds to it. And that shape change causes the pore, pulls the pore open. And in this way, it can act as a calcium sensor to open and close the pore. Now, the interesting thing for our point here is that this crystal structure was determined in the presence of a high concentration of calcium. And in fact, calcium is bound, marked by yellow spheres here. Calcium is bound to this structure, so we know it should be in its shape to open the pore. And in fact, if you look at this pore, and you're observant, you'll say, well, he said the pores are similar in all the channels, but you'd see some difference between this pore and the one we've been looking at. And something I didn't tell you before is the pore of the KCSA channel that we've been looking at was in its closed conformation. And this one is in the open conformation. And so now by comparing them, we can get an idea of how a potassium channel can open and close, what that would look like inside the membrane. So now we'll look just at this part of a closed and an open potassium channel. And here is looking through the pore of a potassium channel from inside the cell. If you were imagine yourself inside looking out through the pore, this would be the KCSA, a closed potassium channel, and here is the MTHK, an open potassium channel. You can see that in the closed state, these helices, the inner helices, come very close together. And if the side chains, if I put the side chains here, you would see it would only be about 3.5 angstroms, very narrow, and make a blockage for potassium, not allowing it to go through. But when a channel opens, it opens to more than 10 angstroms diameter. And if we look at the same thing from the side with only 2 subunits, this is the closed configuration, and this is the opened configuration. Closed, opened. It's really a large conformational change, but it's a very simple one. Notice that what's happening here is this inner helix is bending at the point shown in red. If I step back and forth between. And the helix swings out to open the pore. Now, if you align many different potassium channel sequences, whether they have voltage sensors or calcium sensors or other ligand sensors attached to them, you find that most of them have a glycine at this position. And glycine allows an alpha helix to bend. So we call this a gating hinge. And its conservation makes us think that many potassium channels, in fact, gate in this way. And so, when you look at the channel record and see the ionic current turning on and off, we imagine what happens is the channel is opening and closing in the membrane, opening to let the current conduct and closing to stop it. And the voltage sensor or the ligand sensor attached to it allows the channel to respond to a certain stimulus, and the sensor causes this to happen. And that's how the opening and closing is regulated. Now, if we look at the open conformation, imagine the membrane is from here to here. This is very wide, and so a potassium ion diffusing remains fully hydrated. In a sense, the potassium channel thins the membrane just to the length of the selectivity filter. And I say that because if you were a potassium ion diffusing through, you would be essentially in bulk water when trying to cross the membrane, except when you come to the selectivity filter. And that's with a mechanism we talked about, where there's a beautiful structure and a beautiful mechanism to ensure selective and rapid conduction of potassium. And I want to end with the last picture of that selectivity filter. Here in a picture that highlights the coordination of potassium ions. And I think when one looks at this picture, you can't help but marvel at the beauty of nature, even at the atomic scale. And finally, I want to say that this work would not have been possible if it were not for the enthusiastic young scientists coming to work with me from all around the world over the past 15 years. And I want to show you a list of their names, and I wish I could have time to tell you about them all. I just want to mention two important collaborators at Rockefeller University, Brian Chait, mass spectrometrist, and Tom Muir, protein synthetic chemist, in work we've been doing for the past several years. I want to point out a few people who have been very important to the work that I talked about today. Declan Doyle, João Marai Cabral. Here we were quite a few years ago on preparing to go on one of, too numerous to count synchrotron trips to determine the structure of the original potassium channel structure. This is Yufeng Zhou, and this is a recent picture after she knew what the absolute occupancy of potassium ions in the selectivity filter was. This is Yuxing Zhang, who determined the structures of the ligand-gated and voltage-gated channels, but this was long before we knew what those structures were. This is my mentor, Chris Miller, who started me in science and is a continued inspiration for me. And for anybody who wonders how my lab continues to be so productive and so smooth, wants to know how that happens, Alice Lee MacKinnon. Here's the real reason why. And again, I want to thank Rockefeller University, Howard Hughes Medical Institute, and the National Institutes of Health, and also for the synchrotrons, the radiation sources that make this work possible. Thank you very much.